This is part 2 of your neuron cell communication notes from last time. So on the back of your big sheet, you have kind of in the upper left, you have this diagram. And from the other side of the big sheet, we showed you how neurons communicated with other neurons. And then we said that these neurotransmitters that were in these vesicles get released into the synapse, and then they diffuse across the synapse to the other neuron. So just to give you a little bit more specifics, some of these neurotransmitters that go from neuron to neuron, or that are involved in neuron to neuron communication, that you are probably familiar with or have heard of, are dopamine and serotonin. So those are neurotransmitters that are involved in neuron to neuron communication. The one that I want to focus on actually here is called acetylcholine, because this receiving neuron doesn't always have to be a neuron. It can be a neuron, sure, but it could also be a muscle, and it could also be a gland. And actually, muscles will have a negatively charged interior, just like a nerve cell will, and they can also send an impulse down them, just like a nerve cell does. So there are a lot of similarities here between muscle and neurons and how they respond. And the primary neurotransmitter for neuron muscle communication is what's called acetylcholine, otherwise known as ACH for short. And basically, this is going to stimulate muscle contraction. Right. You don't really have to know this, but I'll give it to you anyway, and you might know this if you took anatomy in the past, or maybe you're taking it this year. But basically, what happens in the muscle tissue is that, I'm just going to type this too. So depolarized muscle releases calcium from a structure called the sarcoplasmic reticulum. And remember that calcium is a second messenger, and this basically tells the muscle to contract. So just some extra stuff. And if you wanted to sketch this, again, you don't have to, but muscles have part of their ER, their endoplasmic reticulum, actually contains these calcium ions. So I'll say ER in muscle. So this is kind of like the ER that I drew there. It doesn't really look like ER, but whatever, go with it. So the ER in muscle cells is really the sarcoplasmic reticulum. It's a specialized structure, and that stores calcium. And then actually calcium channels get opened as part of the depolarization of that cell. But that's kind of extra stuff, just kind of fun little extra stuff. But what I do want you to know, if you're just looking for the basics and just what you need to know here, it's that acetylcholine is the neurotransmitter for neurons to muscles. And what we're going to go ahead and do now is we are going to give some more vocab to all this stuff. So again, this kind of space in between the neurons, or the neuron and a muscle, is called the synapse. Remember, these do not touch each other. There is space that these molecules, these neurotransmitters, diffuse through. So that's called the synapse. The cell before the synapse, the one that's synthesizing and releasing the neurotransmitters, is called the presynaptic neuron. The presynaptic, it's before the synapse. And the neuron after the synapse, you can probably guess what it's called, is called the postsynaptic neuron. So just some vocabulary. And last time when we looked at the action potential is on in this neuron, this was an excitatory response. And what that means is that the response coming down this axon that released these neurotransmitters that diffused across the membrane and got into these ligand-gated ion channels and then prompted the exact same reaction in the next cell, it caused that response, right? This cell, the receiving cell, reacted positively. That message continued to be sent. That's what's called an excitatory response. So what we're going to do is we're going to show how that response actually ends up getting reversed, because at the end of this, you have all of these neurotransmitters kind of stuck in these ligand-gated ion channels, right? So how do you get them back out? Otherwise, the response stops, right? And there's no way to reset this. So you have to remove and break down these neurotransmitters in some way. And that's what we're going to talk about here on the back. So I'm going to zoom back in. And let's talk about the two ways this can happen. So neurotransmitters, again, once the neurotransmitter is in the ion, the ligand-gated ion channel, you can degrade them or you can reuptake them. These are your two options, degrading or reuptaking. So let's describe what's happening here. Can I please type? Are you going to let me type? I'll try one more time. There we go. Perfect. So if you degrade these neurotransmitters, enzymes are going to break down neurotransmitters like acetylcholine, if you're talking about the synapse between a neuron and a muscle cell. Or dopamine or serotonin or whatever it is. So enzymes break down neurotransmitters using an enzyme called acetylcholinesterase. And this is, again, if we're talking about muscle cells, then the neurotransmitter was ACH. So acetylcholinesterase breaks down acetylcholine. So that's a long word, but it tells you exactly what it does, right? The other way that this can work is that the neurotransmitter can be reuptaken. It can be absorbed back into the presynaptic neuron. So active transport proteins in the presynaptic neuron reabsorb the neurotransmitters from the synapse. So let's go ahead and see how these two processes work. So remember, you have the action potential coming down the axon. This voltage-gated calcium channel is going to open. Calcium is going to diffuse into the cell, right? And that is going to prompt these, in this case, ACH-filled vesicles to move along cytoskeleton. They're going to fuse with the cell membrane, and they're going to release acetylcholine, ACH in this case, because we're talking about muscle cells over here, into the synapse, right? So these, this side of the membrane in this presynaptic neuron has little channels, protein channels, that can reuptake. So actually, we'll cover reuptaking first. That can reuptake full molecules of ACH and just reuse them, right? Another thing that cells can do is it can degrade them and then reuptake them. So let's just say here is, I'm going to make the little dot bigger. So here's acetylcholine. There are enzymes, again, enzymes that break down these neurotransmitters. So here's acetylcholinesterase. It's going to break acetylcholine, let's just say in half, so that you get two little pieces. And then those two pieces are actively transported. They are pumped using ATP, this is active transport, back into the presynaptic neuron to be recycled, right? So again, two things. And actually, I said that this was a channel, and I was mistaken. This is also a pump. So you would also need ATP for this. But in either case, let's say that reuptaking is number 2, so that would have been up here. You reuptake acetylcholine or whatever the neurotransmitter is in its entirety, or you can break it down. So we'll say number 1 was down here. You can break it down using enzymes and then pump those pieces back into the presynaptic neuron. In either case, you need energy to do this. It's active transport, and you recycle, you remove those neurotransmitters from the synapse, first of all, which stops the signal that was being sent, but then you also reuse the pieces and you recycle the pieces, and you can do this again, right? So that's how you can reuptake material. And let's go over here actually for a second. There are 2 responses that this second neuron can basically receive from these neurotransmitters. The first, again, we've talked about this just a second ago, was excitatory. If the response from the presynaptic neuron continues into the postsynaptic neuron, and the postsynaptic neuron depolarizes, the negatively charged interior goes positive, right? That's depolarization. That's an excitatory response, right? And in this most recent example, we had sodium coming in, right? That was the response when the neurotransmitter, the ligand, binded to this sodium, ligand-gated sodium channel, right? So over here, you can say that this would open sodium channels. This leads to more sodium in the cell, and this causes the postsynaptic neuron to depolarize. And this is what we talked about in the last video. However, you can also cause this signal to stop. You can inhibit, you can stop the response. And in this case, you can cause potassium channels to open, and potassium moves out. So I'll say moves out. Or you can open channels that let negative ions in. You can open channels that let negative ions in. In either case, you are making the interior of the cell more negative, right? It's already, negative 70 millivolts. Right? We're already negative 70 millivolts. So if you make the inside of the cell more negative, then you prevent the cell from ever firing. You prevent the cell from ever sending the action potential, because what we didn't talk about here is this dashed line. Basically, it doesn't matter if you have a little bit of sodium and the membrane slightly depolarizes. That's not good enough. You can't slightly depolarize. You have to hit a threshold. And then once that threshold is reached, boom, then you continue, right? If you don't really get there, then it kind of fizzles out and nothing happens, right? But once you reach this kind of threshold, then the entire thing, goes and the action potential is set. So if you make the membrane even more negative, then you're definitely not going to reach that threshold. And this is why either of these 2 options prevents the signal from continuing into the next cell. Right, so this would actually cause hyperpolarization. It would cause hyperpolarization, right? It would basically send the cell back down into here, and it's certainly not going to fire when it's all the way down there, right? So if we're going to add this drawing, we can go ahead and we can say that, well, let's add one here. One way to inhibit the response, maybe instead of activating a sodium channel, letting sodium in, maybe you activated a chloride channel. and chloride ion came in. Well, chloride's negatively charged, and that's going to make the inside of the cell even more negative, so that would certainly inhibit the signal. Maybe the ligand caused potassium to leave, right? Chloride in or potassium out. In either case, the inside of the cell gets more negative, and that leads to no response, right? So you don't always have to have an excitatory response. Sometimes you can have an inhibitory response instead. It just depends on where you are in the body and what the neurotransmitter is. All right, some examples of how drugs work. Remember, drugs regulate this, this synapse. So off to this side, I'm not going to go over all of these, or I'm not going to write them all in. You're free to write them in if you want, and you can pause the video to do this. Botox, actually we get Botox from bacteria, and it blocks the release of acetylcholine. It blocks the release of acetylcholine. So acetylcholine, ACH, is never released from the presynaptic neuron. And again, Botox basically paralyzes your muscles, so the receiving muscle cell never gets the message from acetylcholine and your muscles never contract. Right? That's how it prevents wrinkles, because when your muscles contract, the wrinkles form on the skin over top of that. So muscle never gets the signal, you never get wrinkles. Ecstasy blocks the reuptake of serotonin. It blocks the reuptake of serotonin, so it hangs out in the synapse and prevents it from getting reabsorbed. So it constantly stimulates the next neuron. And that can lead, it makes you more alert. You get vasoconstriction, but your temperature also rises and you can actually die of overheating. For Parkinson's, you have a lack of dopamine. So we actually prescribe agonist, or we use antagonist drugs to block reuptake of dopamine. Ritalin, it inhibits reuptake of dopamine, and norepinephrine. So it inhibits the reuptake of dopamine and norepinephrine. So that basically causes them to linger and hang out in the synapse for longer. Pesticides tend to degrade acetylcholinesterase. It tends to degrade acetylcholinesterase, which means that it lingers out in here and it stays bound to these receptors, and the muscle is basically continually activated and it never resets, so that can actually lead to paralysis. Lidocaine, if you ever go to the dentist, lidocaine or Novocaine blocks sodium channels. So if you never, let's go back up to this other side of the big sheet, it blocks all of these sodium channels. So if you never have sodium coming in to depolarize the cell, then the action potential never gets sent. It never propagates down the axon, and that nerve signal never, gets sent, and you don't receive it. Your brain never receives it, so you don't perceive the pain. Some other ones here. Batrachotoxin, we can talk about this. So this opens sodium channels permanently. It opens sodium channels permanently, which means they never close. This is also what's used for like poison arrows or poison darts. And LSD stimulates serotonin receptors. Sarin gas, sure, let's add it. Sarin gas blocks acetylcholinesterase. All right, so all of these different drugs are going to impact what's going on here in this junction, in this synapse. Right. On your version of the big sheet, you have some examples down here, I believe, of a glucose cell and a taste receptor cell on your tongue, about salty and sour. So sodium, and I think for salty, and then hydrogen, I think for sour, right? And you have glucose. I don't have that version of your big sheet here, so I can't talk about it, but that's basically just showing you that there is an original cell that's responsible for this signal, because what you can get into if you're not thinking about it is, where is the origin of this ligand, of the ligand that we started with way back when, right? Well, it came from the neuron before it. Okay, well then where did that neurotransmitter come from? Oh, it came from the neuron before it. So what I'm trying to say is that there actually is a starting point for these ligands. It's not like an endless loop that has no explanation. There is a starting point for these ligands. And the point is those cells are sensor cells. They're sensor cells like your taste buds on your tongue. They're sensor cells that are sensitive to glucose. And those sensor cells, and this is what these pictures try to show you, again, not my pictures, but your pictures, are trying to show you that those are the cells that are synthesizing these neurotransmitters, because those neurotransmitters are just proteins, basically. And they send them off into a synapse immediately adjacent to a nerve cell, and that's what starts the whole cycle. That's what starts the whole cycle. So I hope this helped. Again, we'll talk more about this in class, and I will see you next time.